I'm here today with Dr. Eigenmann, and I want to thank you for being here. We will discuss about a recent article you had on the clinical and translational allergy about TSLP, or thymic stromal lymphopoietin, but because this is a mouthful, we might as well call it TSLP. We have known up to now that TSLP is important for asthma and for atopic dermatitis, but you have very recently shown that it is also important for food allergy. What do you believe is the link between TSLP and allergy?
Well, TSLP is an inflammatory protein, and it has been, as you said, well known for quite a lot of time that it's involved in atopic dermatitis. But obviously when you're talking about allergy, you also think about asthma or food allergy. So TSLP is very well known to be involved in atopic dermatitis, but obviously it has been we thought that it could have a role in asthma or in food allergy. And that's why we started to look in this very well-characterized animal model of food allergy, which I think it's fairly easy to look at if TSLP has a role. So this was really the idea of getting to TSLP.
Yes, and a very good result. I see that you have conducted both a clinical evaluation, you have conducted in vitro experiments, you have conducted ex vivo experiments with a very as you have said, a very well characterized model. So would you like to tell us a bit about the approach you have used to give us those results?
Yes, so the model basically does an IgE type sensitization of food allergy by using a food protein and by using an adjuvant which is chlorotoxin, yes, which obviously is not a natural adjuvant, but at the end you get an outcome in the mouse where you have specific IgE to the food, and when you give the food, when you challenge the mouse orally with the food, you get food allergy, you get anaphylaxis. So this is very close to the human outcome, even if the use of chlorotoxin is not very physiological. Obviously.
Oh yes, I know. That's one of the main disadvantages of mice protocols, because they do not develop our allergy.
Exactly.
So yes, I understand. And what have you found? I believe you have found that indeed TSLP is important for food allergy.
So what we did is, first we wanted to see if TSLP plays a role in this model with regard to sensitization and elicitation of anaphylaxis of challenges by using wild-type mice and mice lacking of TSLP receptor. And actually what we found was that the mice which were knockout, they had much less anaphylactic responses than the wild-type mice. That means that they were somehow protected for anaphylaxis. And this, I think, is one of the main findings of the paper, showing that at least in part TSLP is involved in the pathogenesis of food allergy in this mouse model.
Yes, I have seen that you have found that many tough helper 2 cytokines are underexpressed and underproduced in these mice, and many tough helper 1 and tough helper 17 are overexpressed.
Right.
And this was a very important finding. It was interesting to see, however, that the IgE in both knockout mice and the wild-type mice was the same.
Exactly.
So I believe that if people who are not allergists would be startled to see something about an anaphylaxis that does not entail IgE, what do you have to say about that?
Well, we know that IgE, while it's a very specific marker in humans, it's not that clear in mice. And we know now that anaphylaxis in mice is not only IgE mediated. If you look at the other marker for Th2 type responses, which is IgG1 in mice,
yes,
this was much different.
Yes, I have.
So our hypothesis behind that is that, as I said, it's partly responsible. TSLP is partly involved in the food allergic reaction, but it's not the thing which makes the whole difference. And somehow it's probably not linked to the IgE, since, as you said, IgE is at the same level in both mice.
Would you believe that since there is this matter, what do you say about the ability to extrapolate this to humans? Because mainly, of course, mainly humans have an IgE anaphylaxis, whereas you know, as you have already told us, mice and rats have other means of anaphylaxis. Would you believe that humans could also have this means of anaphylaxis, and we can start and move towards humans with our research?
It's a good question. Actually, you're right. I don't know. Maybe this is something that you see only in mice and has absolutely no relevance in humans. Now, there were some studies done with TSLP antagonists and asthma, for example.
Of course.
And I think at this stage, at some point, it might be interesting to start to look at humans, maybe first in an in vitro model somehow. We would have to think of how we can design something like that. But you know that food allergy, as asthma or other types of allergy, it's probably not only one mechanism. If we can find that TSLP plays a role in human, it would be a way to somehow modulate food allergy, because we are desperately in need of a treatment of food allergy.
Oh, we know we don't have a treatment for food allergy, and food allergy is a constellation of things, and apparently we have just saw that TSLP is one of those things.
Right.
But other than the extrapolation issue, and because I've had my first share of rodent models in my research work, I know that when you're dealing with rodents, there could be some problems. Obviously, when you're dealing with humans, there could be more problems, like, you know, consent forms drop out. But when you're dealing with mice, there could be some problems. Did you have any problems? Do you believe that your study has some limitations that have to do maybe with the model protocol or any other limitation?
Well, the main limitation is probably what we discussed earlier, which is the use of chlorotoxin. Because obviously chlorotoxin is not something which is physiological and you wouldn't use in humans. So I would say this is probably the main limitation. Otherwise, I think the big plus of the animal model is that at the end you're able to sacrifice the mice, you're able to get cells from the gut, you're able to stimulate them for cytokine production. So this is probably, I would say, the main limitation. This is also why we did then the second part of the study, where we looked at the role of chlorotoxin on TSLP.
Yes, you did the second part, and you did not use any beta-lactoglobulin then. You just used the cholera toxin. As I understand it, you tried to mitigate this problem, right. And you did that very well. So it was indeed a very important article, because it showed quite clearly that when there is no TSLP, we have less tough helper 2 cytokines and more tough helper 1 and tough helper 17. And I think this will help us, this will put a base so that we can continue with the research. Do you intend to look into other pathways of anaphylaxis, for example, IgG1 or the complement system in your future research?
We did not plan to do that, but there are other groups who look at that. There is an American group from Fred Finkelman who did quite a lot of work about intestinal anaphylaxis, and I think they defined very nicely that part of the anaphylactic reaction, at least in mice, is not IgE dependent. So there were quite a lot of studies done already until now, but there is still much to do about food allergy so that we understand exactly what's going on.
And I think this is a good base so that we can build upon this for our research to come. Thank you very much, Dr. Eigenmann.
It was my pleasure. Thank you.